Bismillahirrahmanirrahim. This is the second video on the March 2022 paper 2.2, which I have just completed it, and that will be uploaded in a little while. This is the second video, so we're starting with question number 5. Control of heartbeat is myogenic. Myogenic means the heart muscle can initiate its own impulses, while other muscles in our body need motor neurons, but this can initiate its own impulses. Like I say, it has got its own starter, like you have a starter in the car, and you put your key in it and you start the car. This means the electrical activity controlling the rhythm of a regular heartbeat begins in the heart muscle itself. Atrial fibrillation (AF) is an abnormal heart rhythm that causes rapid and irregular contractions of the atria. Untreated cases of atrial fibrillation can lead to stroke. Stroke is a burst or a blocked or a damaged artery in which the blood starts to leak and it results in a brain hemorrhage. So can lead to stroke. A stroke is caused when a small blood clot, often forming in the left atrium, is carried by the blood to the brain where it blocks a small artery that leads to brain damage. List all the structures through which a blood clot in the left atrium must travel to reach the blood vessels supplying the brain. The structures must be listed in the correct sequence. Actually, if the blood clot is in the left atrium, so let's look at the diagram of the heart. This is the right side, this is the left side, this is the left atrium. So if a clot is here, it has to pass through the bicuspid valve. This is the bicuspid valve. From the bicuspid valve, it goes into the left ventricle. From the left ventricle, then out comes the aorta, and there's a semilunar valve here. So bicuspid valve, left ventricle, semilunar valve, or it's also called the aortic valve, and then it enters the aorta, and the aorta forms an arch, and then there are going to be blood vessels or arteries which are going to the brain. So you have to give me bicuspid valve, left atrium, semilunar valve, and the aorta. And this was only for 1 mark. This wasn't, so all the 4 should have been present: bicuspid valve, left ventricle, semilunar or aortic valve, and the aorta. Then next part of the question, explain why blocking a small artery in the brain leads to brain damage. Basically, you've got to understand, what does the blood bring to any part of the body? An artery is blocked, that means that it's not going to carry, the blood is not going to reach the arteriole and then the capillaries and all. So the blood brings glucose and oxygen for basically aerobic respiration and the release of energy. So if glucose and oxygen is not going to reach a certain part of the brain, then of course that part of the brain cells is going to be damaged. So glucose, oxygen not reaching the brain cells, that part, just like the coronary arteries get blocked. So a part of the heart muscle does not receive glucose and oxygen, so that part of the heart muscle does not respire. So if there's a small portion, that's going to die. But if it's a large portion, the person has a massive heart attack and he's not going to survive. Now coming to the B part of the question, a common cause of AF, which is atrial fibrillation, is when a small group of muscle cells in the wall of the left atrium starts to send out electrical impulses to the surrounding heart muscle cells. Explain how the control of heartbeat by the sinoatrial node can be disrupted by atrial fibrillation, resulting in rapid and irregular atrial contractions. Now that's a little difficult question, because it makes you think. If you know the whole heart biology and physiology, only then you can do this. As you can see, the mark schemes are very simple. All this is going to result in extra impulses from 2 different locations. Now we suddenly have a portion of the heart muscle which is also initiating impulses. Now the SAN is in the wall of the right atrium. Impulses interfere with each other. Excitation spreads over both the atria. Now this will result in irregular, since loss of SAN control. The sinoatrial node has lost its control. Now there's another also which is initiating impulses. So this becomes an irregular heartbeat since there's a loss of SAN control. And rapid, why is it rapid? Because there are extra contractions. So why the heart rate is disrupted, resulting in rapid and irregular atrial contractions. So irregular since there's a loss of SAN control, and they're rapid since they're extra contractions. Because it's not only the SAN, now we've got another boss as well. we've got another portion of the heart muscle which is initiating impulses. So that is why there's going to be rapid and irregular atrial contractions. Now coming to the part C, red blood cells are involved in the transport of oxygen and carbon dioxide in the blood. Figure 5.1 is a diagram representing the exchange of oxygen and carbon dioxide between a red blood cell in a capillary and a respiring cell. Some of the reactions that take place in the red blood cell are also thin. The diagram is not drawn to scale. So we have the basement membrane, capillary wall, endothelial cells. We have a red blood cell in the middle. We have CO2, H2O, enzyme X, H2CO3, carbonic acid, dissociates into hydrogen ions, bicarbonate ions. The hydrogen ions attach to hemoglobin, and then the oxyhemoglobin forms hemoglobinic acid or whatever, and then the oxyhemoglobin gives us the 4O2 and the Y. So we've got enzyme X and we've got Y. Now it says identify enzyme X and molecule Y. So what is enzyme X and molecule Y? Now as you can see, enzyme X has to be carbonic anhydrase. Now this is in the new syllabus, this is specifically mentioned that you got to know this. But then you see here hemoglobin, oxyhemoglobin and hydrogen ions, and the oxygen is given away. So what is this Y? This is HHB. What is HHB? Hemoglobinic acid. So Y is hemoglobinic acid. If you see my original video on this, this is something which I have very clearly explained in that. Then it says the hydrogen carbonate ions shown in figure 5.1 leave the red blood cell and are replaced by chloride ions. State why it is necessary for chloride ions to enter the red blood cell as hydrogen carbonate ions leave. But the important thing is the negative ions entering an RBC replaces a negative ion. So maintaining electrical neutrality of the cell. If the electrical neutrality is maintained, only then, otherwise they'll become charged and they'll repel each other. So to maintain electrical neutrality of the cell, or you could have said negative ions entering the red blood cell replace a negative ion leaving, or you could have said reducing charge separation across the membrane. So that's all what you could have said. Then coming to the next part, identify the aqueous environment labeled Z in figure 5.1 that surrounds the respiring cell. Everybody knows that, tissue fluid. Anything surrounding the cell is tissue fluid. 1 mark. Then coming to the E part of the question, oxygen and carbon dioxide are also exchanged between blood capillaries and alveoli in the lungs. The gas exchange system has specialized cells to prevent harmful microscopic particles that are present in inhaled air from reaching the alveoli. These particles are associated with many respiratory diseases. Explain how specialized cells in the gas exchange system prevent harmful microscopic particles from reaching the alveoli. 3 marks. Now, gas exchange system prevents harmful microscopic particles from reaching the alveoli. You know there is this whole system which goblet cells produce mucus, so production of mucus by the goblet cells. And the mucus is a sticky thing, so the whole trachea and the inside of the air passages become sticky. So when the air enters, when you inhale, the air might contain dust particles which you can't see, of course, or bacteria or viruses, and those will stick to the mucus. So mucus is sticky and traps the particles of dust. Then there is cilia inside. We've got ciliated epithelium, and these cilia are going to move, and they move the mucus. They do a sort of a movement by which they brush the mucus upwards, and the mucus is carried up to the throat where it is swallowed. So the mucus is plus the dust particles and the bacteria and the virus, all these things are carried up to so that they're swept up and they reach the throat, and here you swallow it. It goes into your esophagus and then it goes into your stomach. But in the stomach you have HCl and pepsin, which is going to destroy it. But it will not reach, these particles and these bacteria will not reach your alveoli. So you will not suffer from any infections of the lungs. So cilia move the mucus, and this mucus with the dust particles is moved away from the lungs because it's taken to the throat, and here you swallow it because In your just behind the trachea is the esophagus. If you look at a section of your neck, in the neck you have the trachea in front and the esophagus at the back. So when it is moved upwards, the mucus as it enters the throat, then we automatically swallow it. And then, of course, it goes into the stomach, and then the HCl and the pepsin are going to destroy it in the stomach. But your alveoli are saved from any damage. Now, coming to question 6, which is a very good question because it is examining the new part of the syllabus. Figure 6.1 is a diagram showing the structure of part of a DNA molecule. And they've given you this molecule and they say the dotted lines are the hydrogen bonds. Identify structure E and structure F. So you have to identify structure E and structure F. Now what is E? E is deoxyribose. You can't say pentose because it said identify. When I ask you your name, you tell me your name. Your name is Ibrahim. But if you don't say I'm a boy, you say your name is Ibrahim. So identify structure E and structure F in figure 6.1. So E is deoxyribose and F, what is F? The thing you've got to understand is F has to be, it is a single ring. So what is it? It's a pyrimidine. Double ring is purines. So you have to remember adenine and guanine are purines. And there's a U in it, they are double rings. So if this is adenine, what have you to understand? Then this has to be guanine. So F has to be cytosine. Because it says here in the question, it says diagram structure of a part of a DNA molecule. So adenine pairs with thymine. And if this is guanine, then this has to be cytosine. So one hydrogen bond, there are three hydrogen bonds between cytosine and guanine. So identify structure E and structure F. So structure E is deoxyribose. And the other is cytosine. Then on figure 6.1, draw a circle around one nucleotide. Now you've got to be understanding what is a nucleotide. A nucleotide is a 5-carbon deoxyribose plus the base. So it can be this. This is a nucleotide. Or you can do a whole lot of nucleotides. I'm not saying you can't do this because it has to be the carbon 1 is the base to carbon 2, carbon 3, carbon 4 and carbon 5. So this is the phosphate which is attached to it. So nucleotide have to be this, could be others one also could be labeled, could be correct as well. So it's not necessarily only going to be a ring drawn on one base, one deoxyribose and one phosphate on the same strand and passing through the hydrogen bonds. So you have to do a circle, this is one mark, then state the name of the covalent bond that links two nucleotides together. Now what is that bond? That's called a phosphodiester bond. That is something that we keep on telling you. Phosphodiester bond. Then coming to the B part of the question, figure 6.2 shows the RNA base sequence of a short length of primary transcript. Primary transcript. DNA base sequence used to form the primary transcript. So this, as you know, has got U in it. So this is the pre-mRNA. And this is the base sequence using. So what would be of it? So this would be CCA. Then it would be CGA. Then for AAU it will be TTA. And here it will be GA and T. There is only one mark for that. Sadly, if you got that wrong. In eukaryotic cells, the primary transcript is modified to form mRNA. Explain how the primary transcript is modified. Primary transcript is modified to form RNA. This is now the new syllabus. This was not be in the old syllabus. So this is a question on the new syllabus. So the C part in eukaryotic cells, the primary transcript is modified to form mRNA. Explain how the primary transcript is modified. Removal of the introns. So the introns have to be removed and joining together of the exons and the coding sequences are joined. Or you could have said splicing or joining together of the exons and the coding sequences are joined. Then coming to the D part of the question, the mRNA strand is translated at the ribosome to form a polypeptide. Describe how the process of translation results in the formation of a polypeptide. That's 5 marks. It's a lot of marks. And this is the last part of the question and the last question in the exam. So what are we going to talk about? We're going to talk about the mRNA binds to a ribosome. 2 codons at a time are exposed to the large subunit. A tRNA codon attaches to the first mRNA codon. Complementary base pairing occurs. Specific tRNA brings a specific amino acid. A second tRNA brings another amino acid. Peptide bond formation between the two amino acids. Then you give me details like it's a condensation reaction. Then ribosomes move along the mRNA and the next codon is read. The first tRNA leaves the ribosomes. Or you could have given me a little more information. The first codon is always AUG, or the first anticodon is always UAC. First amino acid is always methionine. Ribosome moves along the mRNA in a 5'3" direction. And then the enzyme peptidal transferase, which results in the peptide bond being formed. And then eventually there's a stop codon which is reached and translation stops. Now you can see this all in writing. mRNA binds to a ribosome. 2 codons at a time are exposed. A tRNA anticodon attaches to the first mRNA codon. Complementary base pairing occurs. A specific tRNA brings a specific amino acid. A second tRNA brings another amino acid. Peptide formed between 2 amino acids. This is a condensation reaction. Then further there are quite a few other points as well. Ribosome moves along the mRNA and next codon is read. The first tRNA leaves the ribosome. First codon is always AUG. First anticodon is always UAC. You can see how this is, you can see this. First amino acid is always methionine, MET. Ribosome moves along mRNA in a 5' to 3' direction. Peptidal transferase. So peptidal transferase forms the peptide bond, the enzyme needed to form the peptide bond when the amino acids are brought together. Eventually a stop codon is reached and this results in the translation being stopped. So transcription has taken place in the nucleus, translation takes place on the ribosome, and when a stop codon is reached, and that is going to be the end of it, and the polypeptide chain is formed, and then of course the folding is automatic because the R groups dictate the folding. If there is sulfur, then another down the road there's another sulfur amino acid, so there's going to be a disulfide bond. So basically once the primary structure, which is the sequence of amino acids, is put together, then the folding is all automatic because the R groups then dictate whether hydrogen bonds are going to form or ionic bonds are going to form or disulfide or van der Waals or hydrophobic hydrophilic interactions are going to take place. That finishes this paper, which is the March 22 paper, which was one of the latest paper. Of course, the result has still not come out of that exam, but we've managed to do it, and I have just discussed it with you, and I hope this is going to be helpful, and best of luck. This video contains question 5 and 6, which have been explained, and this is the March 22 paper 2-2, which has been discussed. Thank you.